Okay, so sekarang ni I need to cover about the topics of expressions of biological information. Okay, for the expressions of biological informations. Okay, this topic important because you have to do assignment, and this assignment cover about 25% from your carry mark. 20% from your carry mark. Okay, 10% from your UPS and 30% from your lab test. Okay, first we cover about the central dogma of molecular biology. Okay, central dogma. Okay, central dogma we refers about the flows of the genetic informations means from the DNA. Okay, from the DNA that presents in the nucleus, and then this DNA will be transcribed, okay, to form a single strand of the mRNA, and after that this mRNA will be translated, this transcription process, and this is translation process. Okay, translation process occurs in the cytoplasm. This occurs in the nucleus. Okay, this mRNA will move up to the cytoplasms and undergo process translations involving ribosome, okay, to form a sequence of the amino acid, and this amino acid sequence of amino acid we call as polypeptide. Okay, and this polypeptide will be transported to the Golgi apparatus to modify into a functional protein. Okay, so remember back apa yang you dah belajar masa SPM. Okay, and then so another process is this we call as this process we call as protein synthesis. We call as protein synthesis. Okay, so another process is from the strands of the DNA will be replicate to produce another strands of the DNA. Okay, this is we call as replication, and this replication process berlaku or occur during S phase of interphase. So remember back S phase or interphase. Okay, kalau both of you remember what you was learned in school, okay, so during interphase, okay, during cell division, so interphase have 3 stages, where is G1, S and G2. Okay, S refer to the replications of DNA. Okay, so replications of DNA maknanya the DNA is doubling. Okay, DNA presenting at the chromosome, jadi single chromosome, and after the replication process, so the chromosome will form double strands of the DNA and they form sister chromatid. Okay, then kita ada eksperimen. Okay, refer to your lecture note. Okay, we have experiment. Okay, experiment to prove how the DNA replications occur. Okay, one of the central dogma we mentioned about the replication process. Okay, so DNA replications The experiment do by the Messelson and Stark. Okay, Messelson and Stark do experiment, and before they do the experiment, they have 3 hypotheses. Okay, first hypothesis is okay. Okay, they have hypothesis. They have hypothesis. First is mentions about the conservative, semi-conservative, and dispersive. Okay, so tulisan saya nak cepat ni, so you all boleh refer dekat nota kuliah. Okay, so for the conservative, they refer about the parental strands of the DNA. This is parental strands of DNA. Okay, all have parental strands of the DNA. Okay, for the conservative, okay, both strands will be separate. Okay, and after that, the parental strand will be rejoined. Okay, and form another new strands of DNA. Okay, this is conservative. Okay, for semi-conservative, mentions about both parental strands will be separate. Okay, and then both will act as a template by the ends. Okay, after adds with the new nucleotide. Okay, they will produce 2 identical copies of DNA, which consists of whole and new strands. Okay, this is for conservative. Okay, and for the dispersive, mentions about the ends of the replication process. Mention that consists of, okay, they get 2 daughter cells, same like the others, and for both strands consist mix of old strand and new strand, be like this. Okay, okay, any problems you will ask me. Okay, so they do the experiment. Okay, kalau kita refer to the lecture note, okay, from the experiment, Messelson and Stark use 2 material, heavy isotope. Okay, heavy isotope, nitrogen 15. Okay, they have N15, and they have N14. Okay, so means that at the ends of the experiment, they considered that for the conservative, okay, for the N15 it is a heavy change, but N14 is light change. The red one is a N14. Okay, N14, N15 refer to the nitrogenous base that we treat with the radioactive, N15 and N14. And both of these radioactive differentiate based on their density. Okay, so the N15 more dense compared with the N14. Okay, so in the hypothesis, Messelson and Stark considered as have 2 layers of TN, extracts of DNA. Okay, and then for the conservative, they have, okay, mixed layers of DNA. Okay, this one pun we have mixed layers of DNA. Okay, and for the next generation, supposed to be for the conservative, sorry, for the semi-conservative. Okay, they will have mix and new layers. Okay, while for the dispersive, sebab kita ada same antara dispersive and semi-conservative. Okay, so for the dispersive, they will produce, okay, the layers or mixed layer will become thicker. Okay. Okay, after Masterson and start do the experiment. Okay, you all boleh refer diagrams in the lecture note. Okay, first bacteria cultured in the mediums containing N15, so mean the old one. Okay, and then after that extract the DNA. So they find out ada one layer, the density layer at the lower part of the test tube. And after 20 minutes, okay, the bacteria transferred to the medium containing N14. And after it extract, after 40 minutes, after 20 minute samples, they find out the first generations containing all the intermediate, means they have mix of the DNA. Okay, means dia dapat yang ini dan yang ini. Okay, so they will reject the conservative. Okay, and you wait for the sample after 40 minutes and they find out produce two layer, not this layer. Okay, not the only one layer, but they produce two layer. Okay, so they accept the DNA replications occurs in semi-conservative. Okay, in semi-conservative, where is the parental strand will be separate. Okay, separate. Okay, and act as a template. Okay, and finally, it's produced two identical copies of DNA, which consists of old strands and new strands. Okay, nanti kita cover about the process of DNA replication.